Good morning, everyone, and welcome to the second sessions of this morning, of this second day of the PKD-RRC meeting. My name is Alessandra Bolletta. I work in Milan, Italy, and today I'm co-chairing this session with my colleague, Laura. I think she will introduce herself.
Good morning. My name is Laura Onuchik. I'm a postdoctoral associate at Michael Kaplan's laboratory here at Yale.
Excellent. So great to share this with you. So today we're going to have two speakers. So the title is Hot Takes in the Energy Utilization. And the discussion is going to be at the end. Please place your questions into the chat so that we can then collect them. And I would like to start by introducing the first speaker. So it's a great pleasure to introduce Michael Kaplan. I'm not sure he needs an introduction really, but briefly, he's the CNH Long Professor and he's chair of the Department of Cellular and Molecular Physiology, and he's the professor of cell biology at the Yale University School of Medicine. He got his undergraduate degree from Harvard, and then he moved to Yale for his MD Ph.D., working initially with the Nobel laureate George Palade, and then rising up the ranks in that institution. He received a number of awards throughout his career, so it's a long list to read, actually. Work in his lab addresses the ways in which epithelial cells communicate in the kidney specifically, and he's interested in understanding the mechanism responsible for polycystic kidney disease and developing new therapies. So Michael, the stage is yours.
Thank you very much, Alessandra, for that wonderful introduction. I have to say that it is quite daunting to be introduced by both you and Alessandra for this meeting, since in fact, Alessandra, you are the person who developed the field that I'm going to be talking about, and all of the work on this project that has been done in my lab was done by Laura. Can people see my slides?
No, we can't, Michael.
Okay, let me see. Is that better?
No.
Okay, I'll keep clicking. That way? No?
No, we don't see your slides.
How about that way? Is that working?
No.
Huh.
Are you sharing screen, Michael?
That's a good point. Maybe that would be a good thing to try. Sorry about that.
So is Michael a host now?
Yeah. There we go.
Just projection mode and you are ready to start.
Okay, is that good?
So, yes.
Okay, sorry. So again, thank you, Alessandra and Laura. And again, the work that Alessandra has really laid the groundwork for the idea of looking at metabolism and energetics in the field of PKD. And I'm going to hopefully be able to tell you about a lot of exciting work that Laura has done in my laboratory. So the title of my talk is Listening In on the Conversation Between Polycystins and Mitochondria in Polycystic Kidney Disease. I'm sure most of you are familiar with the proteins that I'm going to be talking about. I'm going to focus in my talk on polycystin 1, and most of you are at least as familiar as I am with this protein. It is a huge 4,302 amino acid miserable membrane protein with 11 transmembrane domains, a 3,000 amino acid extracellular domain, and a short C-terminal tail of about 200 amino acids. Feng Chan and Greg Germino some years ago showed that polycystin 1 undergoes an autocatalytic cleavage at a GPS, or G protein coupled receptor proteolytic site. And this was a truly seminal observation that's opened a tremendous number of lines of research into both the trafficking and function of the polycystins. We showed also several years ago, Veronique Chauvet, who was a wonderful postdoc in the lab, showed that the C-terminal tail of polycystin 1 could be cleaved. We still don't know exactly where or by what enzyme, but Veronique showed that there was a sequence that we refer to as an NLS, or a nuclear localization sequence, in that C-terminus by virtue of which this cleaved C-terminus can enter the nucleus. As I will show you in a little while, Greg Germino's group has since shown, and we've seen as well, that this nuclear localization sequence can also function as a mitochondrial localization sequence. And Jim Calvert and Steve Parnell have shown that this nuclear localization sequence can also function as a G protein binding site. So it's clearly a critically important sequence that can subserve a number of functions. Thomas Vimes's group has also shown a smaller cleavage fragment that can also be targeted to the nucleus. And we still don't understand what regulates the distinct cleavages that produce these diverse fragments. What we do know is that many signaling pathways are perturbed in the context of polycystic kidney disease. Basically, any signaling pathway that you can imagine and look for in a cell that manifests deleted polycystin 1 has perturbed signaling in any pathway you can think of, including cyclic AMP, calcium, mTOR, G protein signaling, Wnt signaling, proliferative inflammatory pathways, epigenetic pathways, and we've heard some elegant research on a number of these topics yesterday and today. And this is a sketch from a fairly recent Torres and Harris review with some of the many arrows that have been discovered as being related to signaling pathways that could be involved in PKD. And a more updated version of this diagram would include even more arrows. We don't know which of these arrows are most proximal to the function of polycystins, and we don't know which of these arrows are most proximal to the formation of cysts in the absence of polycystin expression. But about 6 or 7 years ago, Alessandra Boletta and her colleagues made the seminal observation that an additional pathway that should be considered are metabolic pathways. So Alessandra noticed, or Alessandra and her colleagues made an observation that many of us had made as well, but that she had the brains to actually pursue. And that was that cells that lack polycystins acidify their media more rapidly than wild type cells. And after a large body of very careful research, she and her colleagues concluded that polycystic kidney disease cells, or cells that lack polycystin expression, manifest what's called a Varberg effect. The Varberg effect is characteristic of rapidly proliferating cells, such as those that are found in tumors. And in these rapidly proliferating cells, even in the presence of oxygen, these cells prefer to use glycolysis rather than oxidative phosphorylation to generate most of their energy. So this is so-called aerobic glycolysis. And we largely still don't understand why this occurs in rapidly proliferating cell types, but the teleologic purpose of it could be either that tumor cells tend to exist in hypoxic environments, so relying on non-oxygen dependent pathways for energy generation is adaptive. Also, by not taking advantage of oxidative phosphorylation, the intermediates in the tricarboxylic acid cycle can be scavenged for biosynthesis to produce molecules that help cells grow and proliferate. It's subsequently been shown by a number of groups that interfering with the ability of cells to gain access to high energy metabolites, and especially to glucose, can profoundly limit cystic disease. So the Cheney group showed very nicely that an intervention as simple as calorie restriction can dramatically inhibit cyst growth in the RC mouse model. And Alessandra and her colleagues have gone on to show that 2-deoxyglucose, which is an inhibitor of glycolysis, can also slow down the development of cyst growth. Again, suggesting that interfering with the perturbed energetic pathways provides an access point or vulnerability in the context of polycystic kidney disease. I mentioned again that there are numerous signaling pathways that are perturbed in ADPKD. Some years ago, Vanita Takiar, who was a wonderful MD PhD student in the lab, became interested in common regulation of several of these pathways. And the pathways that I'm circling here are the mTOR pathway, which has been shown to be perturbed in polycystic kidney disease, fluid secretion mediated by CFTR, and of course, cyclic AMP production, which is catalyzed by adenylate cyclase. It turns out that all of these processes are subject to regulation by an enzyme called AMPK, or adenosine monophosphate stimulated protein kinase. AMPK is a regulator of cellular energy, or a sensor and regulator of cellular energy. When ATP levels fall, AMP levels rise, and that AMP binds to the non-catalytic gamma subunit of AMPK, leading to a conformational change that makes the alpha subunit a substrate for phosphorylation by upstream kinases like LKB1 or CAM kinase, and the activated AMPK turns off energy utilizing processes, including ion transport, cell growth, fatty acid synthesis, and turns on energy generating pathways like glucose uptake and glycolysis. It's been shown that AMPK inhibits the mTOR pathway, and Ken Hallows showed that AMPK phosphorylates and inhibits CFTR, and AMPK can also inhibit adenylate cyclase. So Vanita conducted studies in the lab to determine whether activating AMPK could lead to inhibition of cystic disease in mouse models. And those data were published about 10 years ago and showed that, in fact, activators of AMPK showed promise in the context of aggressive mouse models of PKD. The activator that we chose to use was metformin. Metformin is an inhibitor of mitochondrial complex 1, and as such leads to inhibition of ATP synthesis and elevated AMP production and AMPK activation as an indirect consequence. Subsequently, it's been shown in the orthologous pig model that the data that we identified in mice are nicely recapitulated. Here, a group in China using the PKD pig model showed that treatment either with 2-deoxyglucose or with metformin or with both together dramatically reversed the phenotype observed in this orthologous PKD model. And importantly, 2-deoxyglucose and metformin and the combination led to activation of AMPK, as revealed here by a demonstration of AMPK phosphorylation. So these data suggest that small molecules like metformin or other AMPK activators, and I should point out that York Pei has done really nice research along with Doreen Peters and their colleagues showing that salicylate can similarly activate AMPK and suppress cystogenesis. And this suggests that activators of AMPK may constitute promising potential therapeutics, and there have been now a few small clinical trials of AMPK activator metformin, suggesting that they're safe. And while these trials weren't powered to show efficacy, it will be exciting to test such molecules in future larger trials. But taken together, what I've shown you so far hopefully convinces you that energetic pathways are interesting targets for PKD, that things that reduce glycolysis, like food restriction, 2-deoxyglucose, can also activate AMPK, as can indirect and direct activators of AMPK. And together, these might provide interesting pharmacologic targets for very tolerable small molecules or approaches to treat polycystic kidney disease. And it's very possible that the exciting work from the Vimes lab looking at the ketogenic diet might also produce its effects, at least in part, through AMPK as a common final mediator. But what we still don't know is whether and how the polycystins directly modulate mitochondrial activity and energy utilization. And here I want to introduce to you a structure in cells called the MAM, or the mitochondrial associated membrane. MAMs are regions of close contact between ER and mitochondria. They actually are beautiful in the kidney. You can see in the proximal tubule and in more distal segments, there are numerous close contacts between ER and mitochondria. It was shown by Kevin Fosquet's group, as well as others, that calcium can depart the endoplasmic reticulum and be taken up directly by mitochondria at MAMS, and that calcium that enters the mitochondria can act as a direct modulator of oxidative phosphorylation. Valeria Padovano, who was a wonderful postdoc in the laboratory, showed that in fact in a mouse model, polycystins localized to MAMS. Here she physically isolated MAMS through a centrifugation protocol. And you can see that polycystin 1 as well as polycystin 2 is present in the MAMS. She went on to show that the absence of polycystins reduced calcium uptake into mitochondria, so that mitochondrial calcium uptake seemed to be stimulated by the presence of polycystins. And she also showed that mitochondrial oxygen utilization and oxidative phosphorylation, here in something called a seahorse assay, is much higher in cells that express polycystins than in cells that don't, suggesting that the polycystins, or a pool of polycystins at least, are present in mitochondria and mediate calcium uptake from endoplasmic reticulum into mitochondria, where it might regulate oxidative phosphorylation pathways. And it's worth noting at this point that Xiaolong Huang and his colleagues published a really interesting and provocative paper in JASN recently, showing that polycystins in the endoplasmic reticulum might facilitate ER calcium release. So I told you that polycystin 1 undergoes cleavages, that the cleaved C-terminal tail can enter the nucleus and can enter mitochondria. And we wondered, actually, this was an experiment that was proposed by Brad Yoder probably about 15 years ago, and I want to acknowledge and thank him for this important contribution. We wondered whether expressing just the C-terminal tail of polycystin 1 in a mouse model of polycystic kidney disease would have any effect on the cystic phenotype. And the work I'm going to show you now was pushed forward by Laura Onuchik, who's done a fabulous job on this project in the lab. What was created was a mouse model in which polycystin 1 was flanked, the gene encoding polycystin 1 was flanked by LOX-P sites, and we made a back transgene that had a C-terminal tail of polycystin 1 construct under the control of a stop flock sequence. In other words, activating CRE, in this case with doxycycline, which activates the PAX8 TED-on CRE, leads to loss of polycystin 1 expression, and at the same time to activation of C-terminal tail expression. So we could ask, is expression of the C-terminal tail sufficient to in any way suppress the cystic phenotype? So very grossly, looking at just 2 kidneys from these animals, this is a kidney from a cystic mouse that does not express the C-terminal tail, this from a littermate that does express the C-terminal tail, and you can see that there is a profound difference in the degree of gross cystogenesis. If we look at a larger cross-section of these animals, here are wild-type animals, here cystic animals that did not receive the C-terminal tail, and here mice that did receive the C-terminal tail. You should note that this N connotes the fact that these experiments were done in a C57 black 6N background, and that's going to be important in a second. Laura went on to show that kidney weight to body weight was suppressed by expressing the C-terminal tail, as were BUN, and as was creatinine. We wondered what protein or proteins at MAMS the polycystin 1 might be interacting with. So Valeria actually initiated a isolation of MAMS and immunoprecipitated polycystin 1 from that fraction, taking advantage of a back transgenic mouse created in the Somlo lab that expresses an HA-tagged polycystin 1. She immunoprecipitated polycystin 1 from this isolated MAM fraction, and we performed a mass spec analysis, and really only one protein of mitochondria or MAMS jumped out as a specific interactor, and that was NNT, for nicotinamide nucleotide transhydrogenase. And Greg Germino and his colleagues have very recently seen a very similar result. That is that polycystin-1 and NNT can interact. NNT is an enzyme present in the inner mitochondrial membrane. It catalyzes the transhydrogenation of NADPH from NADH. So in other words, a reducing equivalent is transferred from NADH to NADP to make NADPH. And this actually employs the energy inherent in the mitochondrial proton gradient.
3 minutes.
Okay, thanks. NNT is localized to mitochondria, as is the C-terminal tail construct that Laura used, as well as in nuclei. Very interestingly, we did some literature research and found that C57 black 6J mice, which are widely used in the PKD research field, are spontaneous mutants for NNT. These animals do not express NNT, whereas C57 black 6Ns do. So we wondered, since the C-terminal tail of polycystin-1 interacts with NNT, would the C-terminal tail still rescue the PKD phenotype in a C57 black 6J mouse model? So Laura created the C57 black 6J mouse that expresses the C-terminal tail, and powering through this, she was able to show that expression of the C-terminal tail does not suppress the cystic phenotype in the J mouse. It doesn't reduce kidney weight to body weight, or BUN, or creatinine. Collaborating with Hui Shen at Yale, we looked at metabolomics from C57 black 6N or J cystic mice that express the C-terminal tail. There was no difference in the metabolome in the J mice, but a profound difference in the N mice. And I can't take you through all of the different metabolites, but suffice it to say that expression of the C-terminal tail suppressed many metabolites that have been seen in the context of ADPKD. Finally, we asked, does expression of the C-terminal tail influence NNT activity? So Laura did an NNT kinetic spectrophotometric assay. What she sees is that, of course, J mice don't have any NNT activity. The mice that lack polycystin-1 have reduced NNT activity as compared to wild-type mice, and re-expressing the C-terminal tail produces essentially wild-type levels of activity. We can reproduce this assay in culture using cells derived from PKD minus kidneys, and we can show that expressing the C-terminal tail stimulates the NNT activity, but if we remove the mitochondrial localization sequence, we no longer get this stimulation. So how does this work? Expressing the C-terminal tail in the N mice seems to stimulate NNT activity. And we've considered two possible models for how this might suppress cystogenesis. One is what we'll call the Onuchik model. It's been shown that NNT can work in both directions. It can derive production of NADPH, which can generate reducing equivalents and therefore dampen redox damage. But if it works in the reverse direction, it can generate NADH and therefore consume NADPH. That might actually worsen redox damage and might actually suppress cystic disease by making cystic cells more susceptible to cell death induced by redox damage and therefore eliminating the cystic cells. Another possibility, and this is the model that I initially proposed, but I think Laura is probably right, is that if we stimulate NNT, we're dissipating the proton gradient, and that might lead to less ATP generation, which might lead to AMPK activation. So going forward, we're very excited about exploring these possibilities and also exploring the possibility that delivering this small fragment of the C-terminal tail of polycystin-1 might open the door to gene or peptide therapy, and that NNT might be an interesting disease target for therapeutic development. These are the people who did the work. Hopefully I mentioned their names as I went along. And this is what the group looked like on a good day. And thank you very much for your attention.
Thank you, Michael, for such a great talk. I think we will keep the questions for the end of the session, but please do add them to the chat box. So moving on with the session, I'd like to introduce Dr. Samir Parikh. Dr. Parikh is Professor of Medicine, Professor of Pharmacology, and Chief of the Division of Nephrology at the University of Texas Southwestern Medical School. Dr. Parikh research is focused on mechanisms underlying acute kidney injury and sepsis, and his laboratory has really made incredible contributions to the field, such as identifying mitochondrial maintenance via PGC1 alpha and NAD as novel pathways for resilience against acute physiological stressors. His work has been published in several high-impact journals, and his accomplishments have been recognized with multiple awards, including the Don W. Selding Young Investigator Award from the American Society of Nephrology and the American Heart Association. I myself have had the privilege to work a little bit with Dr. Parikh over the past few months in the AKI Now committee from the American Society of Nephrology, and he's been really an incredible person to learn from. So Dr. Parikh's talk today is entitled Harnessing NAD to Restore Energy Metabolism in AKI. And without further ado, I'll invite Dr. Parikh to share his work with you today.
Laura, can you hear me okay?
Yes, yes.
And slides look okay too. You can see the pointer.
Yeah, perfect.
Perfect. All right. So Laura, thank you so much for that really lovely introduction. And this seems like a good venue to acknowledge the fact that I tried to be a PKD researcher and I failed. My K08 was on experimental models of PKD. And I've quickly figured out that I couldn't compete with the great scientists in this field. And so moved on to study acute kidney injury. And so very glad to have this opportunity to present to this group. Those were my conflicts. None of them are relevant to the talk. Another sort of editorial comment I'll add up front is that one of the joys of being in Dallas has been to learn from outstanding PKD researchers who are taking part in this meeting, Ronak Lakhia and Vishal Patel. And so I'm learning all the time, and what a pleasure to follow Dr. Kaplan. Okay, so here's how I think about acute kidney injury, and I'll try to draw some links to energy metabolism as we move along. Really interested in this phenomenon of organs under stress and how they fail. And so acute kidney injury, it's not a surprise to anyone, is an age-associated phenotype, but maybe what could be a surprise is the magnitude of that risk and how that rises with age. And this has been kind of a captivating idea for me for well over a decade, which is, what is it about a low replication organ like the kidney plus age that equals changing susceptibility to the stressors that so many face. And so we arrived at mitochondria through, a lot of RNA sequencing and different conditions that I won't take you through, but suffice it to say that an initial focus in the laboratory was this gene called PGC1 alpha, which stands for PPAR gamma coactivator 1 alpha. And its main job that had been sort of described 20 years ago now was to coactivate the transcription of genes that become structural and functional components of mitochondria, which is a long way of saying it's a mitochondrial biogenesis activator. And what we found initially was that depletion of PGC1 alpha increased the susceptibility of animals to infection-like stressors, the susceptibility of their kidneys to that. And over time, what we were able to flesh out was that this phenotype in knockout animals extends to totally distinct other kinds of noxious stimuli. And conversely, that if you have gain of function for this PGC1 alpha just in the renal tubule, that's sufficient to confer protection. And so this matrix of sort of 6 experiments really became our beachhead to think about mitochondria in a somewhat more serious way in the context of acute kidney injury. And so, I won't get too much more to biochemistry than this, which is to note that the kidney is at least in the mammalian cortex, is a highly oxidative tissue that behaves sort of similarly to the heart. So It preferentially uses fatty acids, although not exclusively, but preferentially uses fatty acids to generate ATP. And the top part of this is beta oxidation. And where NAD fits in, which I'll talk about shortly, is really in the middle to carry away, a form of hydrogen, which is wholly unnatural and needs larger molecules to stabilize it, which is a proton with 2 electrons on it, right? The hydride ion that we all had to grapple with in organic chemistry. And so that's what NAD's job is to carry away this pair of electrons and deliver it to the electron transport chain. And what we found was that in acute kidney injury, when we did metabolomics on tissues, that there was a really interesting pattern. The storage form of fatty acids was building up in these tubular cells, and ATP was down. And knowing that fatty acids were the chief source of fuel kind of got us interested in what's in the middle, right? And NAD itself was reduced in these acute kidney injury tissues. Interestingly enough, those knockout animals I showed you on the previous slide that are highly susceptible had the same look. The directionality for all 3 of these was the same, even in the absence of acute kidney injury. And conversely, those gain-of-function animals had the opposite look at baseline. And so again, that made us zero in on this NAD in the middle. And because of time, I won't show you all the data behind this, but it turns out that levels of NAD are rate limiting or can become rate limiting for fatty acid oxidation. And that's been experimentally demonstrated by a number of laboratories. So this is not, we're not talking about necessarily the redox function, NAD's conversion to NADH. This is just levels of NAD. What is the NAD availability? And so this made us think that if NAD can become rate limiting when it's shorted, that this could be a problem for the conversion of fatty acids down to ATP. And so thinking again about that PGC data I showed you and trying to put it together with this metabolomics work, we stumbled upon a series of enzymes that I have to admit, even now, sort of proposing these enzymes as having anything to do with anything of importance in the kidney or anywhere else felt like sticking our heads on the chopping block. And I'll show you why in just a moment. Series of 8 enzymes that are responsible for converting tryptophan, essential amino acid, down to NAD. We're all being coordinately regulated by this PGC1 alpha, PPAR gamma coactivator 1 alpha. And when you look at the transcriptional profiles in the kidneys, that coordinate regulation was quite evident. And so here was a mechanism that potentially could link the phenomenon of kidney protection we were seeing with PGC1 alpha with some changes in cellular metabolism, specifically around NAD availability. And so NAD does a lot of things in cells. You heard from Dr. Kaplan about how NAD lives in equilibrium with NADP. And these things have different roles, many different roles. And so NAD is involved in hundreds of redox reactions itself. It is a precursor to form NADP. NADP is involved in signaling as well as oxidative defense. And then NAD can also be, quote unquote, consumed and turned into nicotinamide, which is this little piece right here in the upper left of this dinucleotide. And so we'll talk about this all a little bit more, but I put this out there as a preface to say that once you get to NAD, it doesn't totally lend itself to reductionist approaches because there are so many reactions that this thing is involved in, both as a substrate, as shown up here, as well as a redox cofactor shown vertically. Okay, so we have this complicated pathway, many different enzymes that sequentially convert tryptophan to NAD. And in mammals, it turns out that the most prominent way to generate NAD is actually a much simpler pathway where vitamin B3 in the form of nicotinamide is simply ingested and through one intermediate can become NAD. And so seeing the deficit in the enzymes and the metabolites along this yellow pathway in the setting of acute kidney injury made us wonder if we could sort of sneak in through this back door, which turns out to be the metabolic front door, in models of AKI. And so this is what we found. The green arrow indicates, where we intervened in animals, giving them nicotinamide. What's graphed is over time, the serum creatinine curve. And you can appreciate that even in animals, ischemia reperfusion looks very different from cisplatin injury. And you can see that in these two totally distinct models, nicotinamide was sufficient to largely normalize the response of mice. And I should add that for those PGC knockout animals I showed you a few slides ago, we did the same experiment, and giving nicotinamide normalized the heightened sensitivity of those animals to a variety of stressors. Okay, so how is nicotinamide working, right? I already told you that I don't think it's going to lend itself to reductionism, but we can take some hints from the literature. So normal physiology in the kidney, research going back nearly a century, like I said, implicates fatty acids as a preferred cortical fuel. Conversely, we know that when there's tubular injury, the fat builds up. So Venk's group in San Antonio showed this in the early 1980s. Dick Zager came back and did it across a number of different models and showed this as a conserved feature. And then a number of laboratories have sort of re-demonstrated the role of fat in tubules as potentially a promoter of fibrosis. I put this paper up here towards the end of Hans Krebs' career, illustrious career. He became interested in renal metabolism, and he described medium chain fatty acids as a preferred fuel in the mammalian renal cortex. We observed by mass spec that all these different di and triacylglycerides, so each bar here represents a different mass to charge ratio, they were all elevated in the post-ischemic kidney compared to the sham. And so that made us think that one important function may be in the mitochondria, availability of NAD to help convert fuel substrate into ATP. And this notion of the importance of fat metabolism, of fatty acid oxidation specifically, as a renoprotective feature, at least in these AKI or AKI to CKD models, has been independently confirmed by other groups as well. And so we come to this notion again to think about, this sort of central metabolism happening in normal cortical cells. And, we've done some work to try to elucidate this and try to see if it translates into humans. And I think what's been really gratifying is to see some of that work be recapitulated and extended in new ways. Two different papers independently implicating this de novo pathway, those 8 enzymes I showed you a few slides ago, as being important in kidney phenotypes and being important in whole body NAD homeostasis, that the kidney is one of only 2 organs, the liver being the other one, that engages in this complicated conversion of tryptophan over to NAD. A number of other laboratories have further extended the sort of other core observation that NAD augmentation may be kidney protective in acute kidney injury and chronic kidney disease models. Hemorrhagic shock here, ischemia reperfusion, and cisplatin nephrotoxicity here. Adenine induced, kind of crystal nephropathy, even ureteral obstruction and diabetic kidney disease. So a variety of situations suggesting that restoring NAD metabolism could help potentially restore the health or resiliency of tubular cells in different contexts. Okay, so back to the pathway, and I apologize, one more cut at the biochemistry. Tryptophan here on the left now, NAD all the way over on the right. One of the really cool things as you dig into this pathway is that although it's profligate at the start, it becomes very specific as you go over to the right, to the point where once you get to this dicarboxylic acid called quinolinic acid, or quinolinate for the conjugate base form, this thing is specific for the de novo pathway. There's no other known metabolic fate other than the sequential conversion down to NAD. And so when we did urine metabolomics in AKI, what we found was a tubulopathy pattern, which is what we expected to find. Sugars pouring into the urine, amino acids, fatty acids too. What we didn't totally expect to find, but we're pleased to, was this quinolinate as well. And so this is a volcano plot where the farther you go to the right on the x-axis indicates how abundant it is in this situation, the AKI situation, and the farther up you go on the y-axis indicates how significant the p-value is. Okay, So we took advantage of this quinolinate excess in the urine as a sort of thinking of it loosely as a potential biomarker for this whole de novo pathway, because we can't go in and biopsy all of these AKI patients. And 2 different studies, a small case control study over time, and then a larger cross-sectional study, both implicated excess urinary quinolinate as being both an antecedent, meaning preceding AKI, as well as proportional feature, proportional to clinical phenotypes like the severity of AKI or the need for dialysis or even mortality, as it turned out. And so with that observational data in hand, of course, interest in intervening. And this was Ali Poyen-Mer who led this single center RCT to do the same thing, that backdoor / front door trick of giving gobs and gobs of vitamin B3 among patients at high risk, in this case, undergoing CABG. And what we observed was we could boost NAD precursor levels in the blood. And to the extent that in a very small study, we could look at aspects of kidney injury, at least the signals looked in a favorable direction. And so, more evidence sort of building towards this importance of NAD biosynthesis through this somewhat esoteric pathway. Paper in the New England Journal from a genetics group in Australia, New Zealand, led by Sally Dunwoody, implicating 2 other enzymes in the same pathway, kynureninase and HAA oxidase, and finding that people who are born with loss of function mutations have a high penetrance of renal developmental abnormalities, hypoplasia, renal aplasia, and then demonstrating experimentally that if you sneak in through the back door, in their case, they snuck in with this form of B3 nicotinic acid rather than this form of nicotinamide, that they could rescue the phenotypes of those experimental animals. So the same trick we used, but different precursor. And so to sort of summarize a lot of literature that I've just thrown at you, here's that pathway one more time with all the enzymes in blue. We focused on this enzyme QPRT, which is the enzyme utilizing quinolinic acid. Work from a Swiss group focused on this enzyme, ACMSD, showed largely the same results that we showed. And then the genetics work implicated this pathway in kidney development from the Australia New Zealand group. So finally, then, in the last couple of minutes, I just want to share a little bit about how these signals have kind of recapitulated in the COVID era. And I'll start again with observational. This is an observational study of urine metabolomics from 2 different centers, one in Boston and the other one UAB. I won't take you through the heat maps, but suffice it to say, we were looking at AKI versus no AKI, both sites, case control studies. Surveys of the urine metabolome led to largely similar principal component analyses, largely similar heat maps with many shared metabolites, and a conserved signature for this excess urinary quinolinic acid at both sites. And there are many differences in how we defined AKI versus no AKI, and so I'd refer you to the paper and KI reports. And then, as we did before, observation to intervention, this is a pragmatic study, a prospective study that we undertook early on during COVID, involving in total around 200 patients, where now in patients with established AKI, a group of these patients received B3 in these gargantuan doses. And we looked at a composite endpoint. I'll skip this. This is just to point out that the pragmatic study had similar numbers of patients coming through with COVID and similar severity of illness.
3 minutes.
Yep. So this is the primary outcome we looked at, which was the renal replacement-free survival. So this is a composite of death or the need for dialysis. And what you can appreciate is that the group that received B3 largely looked like it was doing better. And then after adjustments, and you can see all the things we adjusted for down here because it was not a formal RCT, that signal basically persisted. When we did a sensitivity analysis to subtract out all of the patients with stage 1 acute kidney injury, the kind that we would expect to get better of its own accord anyhow. So kind of a dampener, if you will, in this study. If anything, the magnitude of effect became somewhat bigger. So again, going in the right direction, but I will once again point out that this is a small study. You know, I think what you want to do is to have an RCT that's on the order of 400 or so. So still kind of building towards that evidence base. However, I think that between the small RCT and this positive pragmatic study, there are now a number of funded RCTs that are underway in the US and internationally, looking at different kinds of AKI syndromes. And so we will get an answer in patients as we hopefully over the next couple of years. And I think to summarize then, I think mitochondrial energy metabolism is fundamental to renal tubular health. I didn't have a chance to show you some of the human mitochondrial genetics that supports that statement, but I think our work kind of feeds into that larger notion. NAD regulation may be a determinant of outcomes, at least in acute kidney injury, maybe the transition from AKI to CKD as well. And then finally, augmentation of NAD could be right for therapeutic trials in settings that provoke stress. So we're interested in a lot of different things moving forward. Age, as I mentioned up front, continues to be an area that we want to focus in on. And we're really interested in this acute kidney injury to CKD transition as well. And how does NAD fit into that? And so I'm going to stop here, I think. Yes. And once again, thank Laura, thank Dr. Boletta as well, and all of the organizers for a chance to present at this meeting, and happy to take questions with Dr. Kaplan.
Thank you so much, Dr. Parikh, for a wonderful presentation and the wonderful data. I think we invite the other two discussants of the panel, so Greg Germino and Dimitri Maxim, to join the group. Hi, Greg. I can't see Dimitri yet. There you go. So we open the panel for the discussion now. And let's start with the first question by Paul Welling, maybe. Wonderful talk, Dr. Parikh. Do you know what TF is interacting with in PGC1 alpha to control de novo NAD+ generation pathway?
Yeah, Dr. Welling, thanks so much. I don't think we've met before, but I really appreciate the question. Working on it, and hope to have something together if I'm back for this meeting. But so the question, just so everyone else is following along, PGC1 alpha itself doesn't bind directly to DNA. Its main job is really as a co-activator, and so it binds to transcription factors. And there are many transcription factors that it has been associated with, ER, alphas, PPARs, many more. So trying to figure out, which of those transcription factors are most contributory to this particular phenotype of PGC induction is a great question. Thank you.
I have a question for Samir as well. This is Greg. So in your model, you've looked mostly at proximal tubule models of injury, right? And you've kind of focused on the mitochondria there. How about in the other parts of the kidney? Do you have any information on that?
Yeah, I appreciate the question, and I'll point out that the mouse genetic models we've used are pantubule, right? So that's a PAX8 RTTA that is commonly used as a kind of pantubule driver allele. And you're right, the ischemia reperfusion, depending on whom and how much you believe, is either, a loop of Henle, a thin ascending limb, or an S3 proximal tubule. We use that model, we use cisplatin. We've used other models as well, like folic acid, crystallopathy type models, which might be more pan-nephron. And the protection appears to persist. And so, it's really interesting, why on earth does the kidney make the evolutionary investment to have all of these enzymes expressed to turn tryptophan to NAD? What's the point of that? Why does the kidney do that? Why does the liver do that? I don't have an answer for that, but I do find it really intriguing that both of these organs face an onslaught of xenobiotics from the go. And maybe there's something about having multiple pathways to make this really important cofactor that's important to the health of those organs, at least to get to the point of gametogenesis and moving on to the next generation. So that's not an answer to your question, but I think it's a really provocative one.
Thank you.
Maybe I can add on that to both Michael and Dr. Parikh question on this role of NAD+. It would seem that these will be essential for mitochondrial function altogether. So would you think that rescuing the pathway in PKD would also be beneficial somehow? But I believe some niacinamide studies have been conducted and were not so positive. And the question to Michael related to this is, would NNT fit into the picture? How would NNT fit into the picture of this NAD+ regulation?
Maybe I could just take a quick first cut and then turn it over to the professional. Just sort of thinking in a very fuzzy way about PKD cells as kind of sharing phenotypes with cancer cells. One of the areas people in oncology have looked at with regards to the NAD biosynthesis world is actually to inhibit NAD biosynthesis in different ways, not to augment it. So to inhibit the conversion of B3 to NAD, to inhibit this de novo pathway as well. And so to the extent that one thinks of PKD cells in that regards, it totally aligns, I think, with what Dr. Kaplan was describing in his talk, that if any, do you want to perhaps short, whatever might be happening in the mitochondria? And I'll let Dr. Kaplan actually give the professional answer.
Yeah, no, so that was perfect. Thank you. And this gets back to what we call the Onuchik model in the lab, which is probably the most interesting mechanism, which is that in this context, actually exacerbating the potential for oxidative damage in the PKD tubule epithelial cells themselves might make them more susceptible to oxidative cell death and therefore might limit cyst growth. So we don't know whether NAD in that context is a good guy or a bad guy. But if Laura's model is correct, then actually limiting the available of reducing equivalent might actually stem the development of the disease.
So if I could quickly follow up on that question, actually in these initial niacinamide trials in PKD, I think the first one was published in 2015 by Shaga and Claudie's group. They actually looked at at niacinamide as an inhibitor of SIR2 and 1, and therefore leading to an inhibition of the deacetylation of p53 and a little bit of a different pathway of what you touched on. How do you see these playing together? What do you see as the contribution of these different pathways in terms of ADPKD and renal disease?
Yeah, so now we're sort of getting to graduate level or faculty level questions on this NAD pathway. And so just to bring everyone else along, nicotinamide is a precursor to make NAD. Nicotinamide is also an inhibitor of some of those enzymes that consume NAD. So those would include sirtuins, those would include poly-ADP ribose polymerases, PARPs, as well. And so when you give a bunch of nicotinamide, yes, you are boosting NAD levels, but theoretically you are also inhibiting PARPs and sirtuins. So from a scientific question point of view, it becomes hard to separate which of those effects is most dominant here when you use nicotinamide. But some of those studies I showed you from other groups used other versions of nicotinamide that don't carry that same sort of dual action. The reason we worked with nicotinamide for patient studies is because it has the biggest patient safety data associated with it. So with the gargantuan doses we were going to give to very sick patients, safety is super important, which is, obviously we don't think too much about that with mice, but we have to a lot with humans. And it turns out thousands of reported patients from trials have safely ingested gram quantities of nicotinamide for years on end. So that's the ENDIT trial in Lancet and others. And so among the choice of precursors we could use for these human studies, that one seemed to be the safest.
Thank you. Perfect.
But the experimental models do line up with an NAD type of effect because when you sort of draw the circle around all those precursors, only nicotinamide is an inhibitor.
There's a second question from Paul Welling. Michael, nice talk. Given the importance of AMPK in PKD, is there a role for PGC1 alpha in the disease? So this brings about the conjunct answer, I guess.
Yeah, so actually this question really belongs to Vishal Patel, who's done some really nice work showing that PPAR alpha and PGC1 alpha levels seem to be protective in the context of PKD. I think, and I shouldn't give this answer for him, but I think they showed that miR-17 could suppress PPAR alpha and PGC1 alpha, and that treatments including fenfibrate that elevated PGC1 alpha appeared to be protective. Again, fitting in with the theme that increasing mitochondrial activity and capacity for OXPHOS might be protective, although again, for reasons that maybe we don't fully understand.
So if I may follow up with one more question while we wait for people to put more into the chat, with Dr. Parikh. So do you have any idea of in which cell type specifically these de novo synthesis enzymes are expressed, more specifically in the 3 segments of the proximal tubule? I guess there it will be very important.
Yeah, I want to say that single cell sequencing data pretty strongly implicates, PCT1 and 2 as sites for expression of these de novo enzymes.
Mm-hmm.
So I think that it would fit. And I guess the other thing to just sort of follow up on that, how much of the effect is cell autonomous? So we can certainly demonstrate that. And I didn't show any of the cellular data, for time, but there certainly is a cell autonomous effect with these enzymes, but also recognize that these precursors can kind of float around between cells, and this is metabolism. And so, think about the Cori cycle and the way lactate moves around, and it gives you a sense of, so the kidney turns out to be, for example, a net exporter, we believe, a net exporter for nicotinamide to other organs because it can make its own precursors.
Mm-hmm. Very interesting. It turns out that actually glucose is also synthesized quite fairly abundantly from the kidney and released in circulation as well. So it seems to be a recurrent theme. So Vishal is actually thanking Michael Kaplan for answering for him. He's mutated, cannot intervene. I don't know if he can be unmuted. But there is another question: would mitochondrial uncouplers work in PKD? And maybe this question could be for Greg and for Michael together.
So I will say that this is something we've thought about a lot, since metformin in some ways is thought to act like a mitochondrial uncoupler. We are collaborating with Jerry Shulman, whose laboratory has developed some very exciting tissue targetable mitochondrial uncouplers. And Laura is working with people in Jerry's lab to see if there's any therapeutic efficacy. I would say the results are quite preliminary, but I wouldn't rush out and build a biotech around it just yet.
If anything, go otherwise. So, yeah.
Interesting. Very interesting. One more question for Samir. So Samir, when you've looked at NAD plus and NA, kind of nicotinamide, in that context of acute renal injury and other forms of renal injury, how about NAD plus? What's going on in that context of that your injury models?
Sorry, I missed it. NADP or NAD plus?
It's a NADP. Sorry.
NADP. Okay. Yeah. So the NADP this is one of the really interesting things. I mean, because there's some experimental data out there that if you can augment NADPH, now you have the reducing equivalent that can, help generate glutathione and protect against oxidative stress. And so we don't have that experimental data in hand yet, but it is one of the things that we definitely want to look at. This is, again, going back to that there's probably so many things under NAD, and we've necessarily had to zero in on fatty acid oxidation, but I think you're right. I think your hunch is right on that there's going to be a really important role for NADP. And it's a very doable experiment, so.
Thank you.
So maybe one last question to Michael. With this super exciting data with the CTT being able to rescue the phenotype to quite an impressive extent in the N background. So my question relates to possible alternative molecules that are involved in the phenotype in the J background. Is there any complementary or compensatory pathway that can somehow justify why in the J background you have such a severe phenotype?
Yeah, so this is touches on a really important question that we struggle with, which is if the difference between N and J is so profound in the context of expressing the CTT, why do both N and J mice get cystic disease in the absence of the CTT? In other words, if NNT is required for polycystin function, why don't J mice walk around with cysts all the time? And so our answer to this, and it's very similar, Alessandra, to a question that Fong put in the chat, we think that NNT is not a primary downstream effector of polycystin under normal circumstances. We think instead its activity might be a disease modifier in the context of PKD, and that the CTT's ability to modulate its activity or maybe even bias the direction of its catalysis might in some way be a modifier of the disease that can lead to this suppressive effect. And that's something that Laura is working very hard to try and figure out.
Yeah, excellent. And apologies to Fang, I skipped your question. I just missed it. Thank you, Michael, for bringing that up.
Can I just ask one quick question to Samir, since I have him on the screen and wanted to ask him this anyway? So I guess it's a two-part question. One is, in a PGC1 alpha knockout, what do the mitochondria look like in the kidney? Are there fewer mitochondria? Do they have different shape and structure?
Yeah, so buried deep in the supplement of one of our papers was a little bit of mitochondrial phenotyping to look at this. So it's not like you wind up generating these row zero cells. What you wind up with are cells that, at least to our eye, mitochondria are morphologically normal, but quantitatively fewer. The mass is less.
And just out of curiosity, in the beautiful Nature paper that you guys published, are you looking in N or J animals?
Those are J.
So do you think
J all the way in AKI? And it rhymes, so it must be true.
So would it be interesting to look in N animals to see?
Yeah, right. So going back to Greg's question, I mean, this would be one of the ways to start looking at crossover activity with NADP. And so, formally two different ways, right? And or take the J's and knock out NNT and other of these enzymes. So right on.
Excellent. So I think we are reaching the end of our panel discussion. So maybe Laura wants to close the session.
So let's wrap up this session. We're already in time for the lunch break. I'd like to thank my co-chair, Dr. Alessandra Barrera. Also thank our two wonderful speakers today, Michael Kaplan and Samir Parikh, and also our panelists, Dimitri Maxim and Greg Germino, and for such a compelling discussion. And I think we'll resume at 1 p.m. after the lunch break.
I don't know. I emailed Terry real quick. I haven't heard back. But maybe we'll give it to 1:10 if that's okay, because we're a little bit behind schedule here. But I also wanted to thank everybody for a wonderful discussion.
I want to add my thanks, and yeah, Brad, it's fine. So we are adjourning for most everybody, unless you want to go to the trainee session. So if you do, and you didn't get a link, just please email Stacy and we can get it to you.
It's the same link that they're on right now.
Oh, it is? Okay, great.
Yes.
Okay,